Okay, welcome everyone to today's IADS webinar on derived oral stem cells from lab bench to bedside. Please welcome to pardon, I can't hear my sound. Are you using the YouTube now? If yes, please turn off the YouTube. Okay. Okay, so welcome again to today's webinar. It will be on the oral stem cells from lab bench to bedside. Our speakers today is Dr. Bartosz Kempisty from Porcine University of Medical Sciences, and also Dr. Marta. Please welcome our speakers today, and on behalf of the IADS board, I would like to thank you, doctors, for accepting our humble invitation. And you shall start right now.
Okay, so good afternoon, everyone. My name is Bartosz Kempisty. I'm Associate Professor at the Department of Histology and Biology and Department of Anatomy on the Porcine University of Medical Sciences. I'm also visiting professor at Masaryk University, Brno, Czech Republic, in Center of Assisted Reproduction. And when someone asks me about the stem cells, what are the stem cells? I cannot give the proper answer because the stem cells in the compound process and compound problem right now. I think the 21st century is the century of the stem cell biology and application and uses of the stem cells in the preclinical and clinical study. So today I will talk about some main projects we have in our team. I will show some examples of the animal dental project, of the human oral cavity stem cells projects too, and I will show also some examples of the experimental design based on the animal project, animal model, pig model, porcine model, and on the human model too. Then I will talk about our inspiration of human and animal stem cells research, what was our main inspiration, about the stem cells bank and conditioned medium. Is this really a good direction of the project right now? The difficult beginnings of the oral project, of course, and some experimental design of animal oral project. Cutting our teeth, so the human gingival project. Yes, we start from this project 2 years ago. We have a lot of problems with the project as usual, but now we have some good final results. And final conclusion on our future direction. So I will talk about our dreams and goals of the project in the future, of course. So the main experimental question are 3 questions. So are the primary culture system based on animal and human cells a good model for cellular development capability assessment? This is the first question. The second one, does the human and animal cells primary in vitro culture reflects the huge regenerative and reconstructive cellular properties? Is the primary in vitro culture real science or science fiction? Because when we start with the primary culture system, this is not stable condition, this is not stable homogeneous population of the cells in vitro conditions. This is the main problem of the primary culture system. So the third question is, could the analysis of expression pattern of genes become helpful in cellular developmental capability assessment? That's the third question of our main goals for this project for today. So the cell and tissue engineering group's leader is Marta Dyszkiewicz. She is the clinician. She collaborates with my group. She's the leader of my group of the oral cavity project. We also collaborate with the Saliva company in oral cavity research project. So the main research topic, scientific map, so previous studies and future directions we have in our group, in our team. So the research based on animal models. So I told you before, we talk about the animal model because some experiments we cannot do on the human model. So we have to use the animal model, and the pig model is the best one to analyze some physiological condition, genetic properties, and the molecular assays based on the pig model. So the porcine backup post mucosal cells and cheek fascial cells developmental capability. and we are looking for the expression of connexins, for example. So connexins are the markers for the physiological properties of the cell function because they are form some connection between the cells. Then we are looking for the real-time cellular proliferation capability of the cells, because when we start with the primary culture, then we're looking for the proliferation of the cells. We need to have the logarithmic proliferation of the cells in vitro during primary culture. Then we can talk about some physiological properties of the cells. And after that, we are doing the microarray analysis of, for example, Affymetrix for looking for the expression of some gene and looking for the new marker genes for some physiological process of the stem cells in vitro, of course. And the atom force microscopy as a modern and innovative strategy in morphological and molecular science. We use the atom force microscopy in identification of morphology of the cells in vitro, of course. So the main question regarding in vitro cultivation, in vitro culture system, primary culture system, is that can primary in vitro culture mimic the in vivo physiology? This is the main question we have every time, living in the shadow of the 2D and 3D in vivo culture system. And para-screen activity assays, of course, so that the cell culture insert can be helpful in functional assays. Of course, we are looking for the para-screen activity of the cells from the oral cavity, the stem cells from the oral cavity too. And now we talk about the porcine model, so the pig model of the research, so the porcine buccal pouch mucosal cells and the fascial cells primary culture systems, and we are looking for the selected gene and protein expression assays. We are doing also the short and long-term primary culture system using the porcine buccal pouch mucosal cells, and real-time cellular proliferation properties. We are looking for the properties of real-time proliferation of the cells in vitro. And then the human model research. So we are looking for the human oral cells molecular characteristics. We are doing the analysis based on the transcriptomic profile, so the microarrays. Then is advanced the primary culture of human oral cells, starting from the single adult cells. And now we are looking for the differentiational capability of the cells during IVC, so during in vitro culture systems. The beginning of our long-term relationship with the stem cells, so is the stem cell plasticity the new mark of transdifferentiational capability of the cells? That's the main question. And the second question is how the scattered lines of the microarrays bring us closer to the conclusion of the stemness specificity of the stem cells in the culture system, in the primary culture system too. So what is our next target? So physiological properties of oral stem cells. So how to best mimic in vivo conditions? All the time we have the same question. For the purpose of the research on strategy for regenerative and reconstructive medicine. And now some short examples of the molecular assays. We are doing the microarrays every day, actually. So we have some examples of the heat map when we have some two colors. The red one, it means that this is the decreased expression of some genes, and the green one is increased expression of some genes on the heat map. And this is the ontological groups responsible for the proliferation and migration of the cells from the oral cavity of humans. And then we have some string analysis below of the interaction of the genes, some map of the interaction of the genes of the proteins, where the green color it means increased expression, and the red color it means decreased expression of some genes in these signaling pathways. And from the right upper streaming, we have the proliferation of the cells in vitro using the real-time proliferation systems. Human gingival cells primary in vitro culture. Here we have the 13 days of the proliferation. We start from the proliferation assays and we start from the analysis of the proliferation, because when we have the cells in vitro, for the first time, we need to have simple proliferation, simple system of the proliferation of the cells in vitro. And after that, when we have the confluence of the cells, we can talk about the molecular properties, molecular assays, metabolic assays, proteomic assays of the cells in vitro. And then we have also human dental pulp cell culture. It was very difficult to obtain the culture from the dental pulp, but we have right now a lot of tubes with the stem cells from the dental pulp in our bank. So item force microscope observation is the new technique, the new microscopic technique, which show us the surface of the cells in vitro model. So we analyze the cell membrane structure, and I will show you the examples of the microscopic observation later on. But now we can observe the structure of the cells, structure of the cell membrane outside surface of the cells by using the microscopy atomic analysis. And flow cytometry analysis, of course, we use also to identify the stem cells in our population of the cells in vitro. We back to the porcine oral project. Now we have the porcine backup of mucosal cells during the 7 days of proliferation in vitro, and some expression of 3 genes which are markers for the cell proliferation especially. And here we have also the short and long-term cultivation system of porcine backup mucosal cells. We cultivate the cells for the 7 days, and we cultivate the cells for the 13 days. And this is of course, on the left side, the structure of the figure would represent the real-time proliferation of the cells in vitro. And expression from the right side, we have the expression of the genes. We have expression of the connexins, which are the markers of the physiological properties and the function of the cells in vitro too. And molecular assays support some back approach mucosal cells developmental capability, and we have some ontological groups which are the cell adhesion, cell migration, cell proliferation, cell motility, and they represent the physiological properties of the cells in vitro, of porcine back approach mucosal cells in vitro, analysis of the string and interaction of the genes and of the proteins both in the microarrays too. And then we have also some analysis of the microarrays by using the microarrays and ontological groups responsible for wounding, from vasculature development, and wound healing in the porcine backup mucosal cells during long-term primary culture in vitro. And when we start with the culture of the porcine backup mucosal cells, we have heterogeneous population of the cells. We don't have the same simple homogeneous population of the cells in vitro in the primary culture system. We have some adipose cells, we have some fibroblasts, we have some keratinocytes, a lot of mixture of the cells in vitro. So the main question is, what of the cell types we have in the primary culture system when we start with the primary cultivation? Future directions, so our dreams about the stem cell biology, I will be using the IMA model, the porcine model, and using the human model too. So all of our current research is based on the developmental competence of different cell types cultured primary in vitro. Then we would like to future analyze differentiation capability of the cells in primary culture. So we are looking for the differentiation properties of the cells in vitro, because when we start, we have some primary culture system of the primary cells. Then we are looking for in which ways the cells can be differentiated. We would like to estimate transdifferentiation potency of the cells isolated from adult organs and tissue too. So this is the second process we would like to discover using the primary culture system of the pig model. And finally, we would like to acquire stable homogeneous population cultures of the in vitro differentiation cells for further application in regenerative and reconstructive dentistry and medicine. And the transdifferentiational of the stem cell biology and morphological identification of the stem cells in vitro, so using the tool of mathematical analysis and machine learning. So that's the future direction, because we have some examples of the research based on the machine learning, when we identify by using the computer programs morphology of the cells using our culture system of the human and animal models too. And banking of the cells and tissue for the future is the future directions, and our application, of course, too, because as I told you before, we collaborate with some company, with Salivia company, which have its own bank, and we would like to banking our cells and tissue in the future too. Connecting the science and the practice, and we collaborate with the Salivia Institute of Advanced Sciences, and starting from of course, with analysis of the simple light microscopy through the fluorescent or confocal microscopy, and finish our observation using the atomic force microscopy. Here we have examples from the 2D and 3D culture system and atomic force microscopy observation. From the left side, we have the 2D culture system of the human gingival cells. In the middle one, we have a 3D culture system of the gingival cells, and the analysis of on the atomic force microscopy on the right side of the structure and morphology of the cells in vitro. And some processing on the gingival cells in vitro in our labs from the first to the final step by using the homogenization, by using the incubator, by using some enzymes, specific enzymes to have the simple homogeneous population of the cells in vitro. And gingival cells assays, so we start from the 2D culture system and using the flow cytometry identification of the stem cells markers like a CD44 antigen, CD105, and CD90 marker protein for the stem cells using the gingival project. And the dental pulp stem cells and the short processing of the dental pulp cells in the culture system in our labs, also from the first to the 10th stage, the final stage, of course, and some processing of the enzyme, of course, and the centrifugation and the observation of the cells in vitro, which we have in the. Then in the final step, we are putting the cells in the into the incubator and waiting for the final result. Okay, of course. And dental pulp stem cells assays, of course, when you have the cells, when they reach the confluency, full confluency, and looking for the markers of the stem cell by expression by using the flow cytometry, the same markers like CD44, CD105, and CD19 protein marker expression. And so at the end of my presentation, we would like to show what we can share with you, of course, with the cells, of course. So the human and animal models, we have a lot of cells of human models from the gingiva and dental pulp and animal model too, which we can share with you. The primary cultural cells, of course, with the primary derived stable cell lines. We are working right now on the cell lines, stable cell lines, which we can be generated in our lab. Conditioned media, of course, we have also above when you culture the cells primary, we can take the medias above and use them as the conditioning medias. And we can do also the microanalysis and the assays and the analysis. And we can share with you other materials, techniques, and of course our expertise in the molecular assays based on the transcriptomics, proteomics, metabolomics in some cases, and flow cytometry, of course, too. So thank you for my presentation for this part.
And I will continue a bit more, moving on to clinical part. Of course, continue with the same subject, but just moving a bit from the lab to the clinics. So we continue on the oral cavity and its regenerative potential. And the question for some of you who are, for example, dentists, not scientists, is how much do you know the science behind your everyday practice? Because what we can observe is that we should probably, as a dentist, ask a bit more questions. So to be the dentist of the future is not only because you own so much equipment in your dental office, but also because you really know the answers to what role in dental health plays simple biology. So as a dentist, as a practitioner, you will be performing, or you are performing, a lot of procedures in which you should really involve patients' own biology. But of course, first you have to know how does it all work. So this is a very essential subject. So the reality is that the dentistry, since many years, almost perfected skill of replacement. But the question that remains is it really as perfect as it seems? Why don't we engage more tissue's own potential into the process? And the question is what is and what would be really effective in comparison to what we already know. How can we actually involve this simple, in a way simple, of course, biology? Of course, you can find in your own practice, and if you are a practitioner with some experience, if you are doing some of the surgery procedure or endodontic treatment, you of course can notice, or you can already know from your colleagues' experience, that we can, unfortunately, some of our treatment plans are not successful in the end. And of course, it happens in general in whole medicine and dentistry as well. But what we are observing more now probably is that we have a lot of problems and issues coming with the, for example, implant treatment. But what else is, of course, obvious is that not all endodontic treatment is successful. So we are, all of us practitioners, we are experiencing some of failures, and some of them cannot be avoided, but some part probably could be avoided if our knowledge would be a bit wider. And these are just the examples, just to show you that, of course, you can Google much more and check much more scientific publications on these subjects. So these are just examples, just to show you that this is these are current problems that unfortunately are increasing in our practices. So the prevalence of peri-implant mucositis varies between 19 to 65%. So as you can see, it depends on what kind of study are you looking at. And the prevalence of peri-implantitis ranges from 10 to 40%. So it's relatively high, even if you stick to the lower number. If you look at the population, it's still something that worry us as practitioners. So it looks like there is, of course, a lot to think of for the future. How can we avoid it? Of course, some part of it can be avoided with differently planned treatment or slightly different approach. Some of them are very difficult to avoid, and there's also this part based on involving patients' biology. If you know how the biology in oral cavity works, it is definitely something that will help you. If we are talking about stem cells and stemness potential of oral cavity, of course, we are also discussing in general using PRF, PRP, or concentrated growth factors, although they are sort of taken from patients' blood. But of course, they are implemented into the oral cavity. And we can, of course, now confirm that this is something that is part of successful treatment very often. And it is, of course, now next step probably coming from concentrated growth factors, trying to even more engage stemness potential of the oral cavity tissues into the therapy. What do dental stem cells bring to the table? So you can already see that there are some studies that are not preclinical, they already involve patients. And this is an example of one of them, but of course you can find more. Patients with irreversible pulpitis enrolled and monitored for 24 weeks following the transplantation of mesenchymal dental pulp stem cells coming from discarded teeth, expanded, and to stimulate the sort of regeneration of the vitality of the tooth, with good results. As you can see in the end, you can see that there are the cone beam computed tomography demonstrated functional dentin formation in 3 of the 5 patients. There are also research coming from respected scientists based on the concept of involving the potential regenerative potential of the cells coming from the oral cavity into the treatment of peri-implantitis defects, as well in bone grafts and also in temporomandibular joint regeneration and periodontal tissues. So there are a lot of studies going on right now, and hopefully soon there will be also a next step in these therapies involving patients' own own potential, own stem cells. Part of these research currently are also exosomes, microvesicles. Just as you can see, there is also a potential in exosomes that can be introduced into the practice as well, tested in tooth root slices models with dental pulp stem cells, trigger regeneration of dental pulp-like tissue. So this is, of course, observed in vitro conditions, but of course, you can also find studies that are already in vivo condition. And some of our current research projects that, as you can see, they involve also the aspects of oral mucosal epithelial cell cultures, and as well the review on current status of oral derived stem cells that you can also find online, and you can find a lot of information on how to apply them into your practice, what can be probably applied in the future, what are the ethical and legislation rules right now, regulations, and there's also a lot of information on different stem cells derived from oral cavity and exosomes as well. You can also find some of the banks that are banking stem cells already, also coming from the oral cavity. This is an illustration how it is performed in dental office. So you can stem cells can be collected when the teeth are, for example, extracted due to orthodontic reasons, or during or in children, for example, when you are dealing with the patients with milk teeth. Also in adult patients, when you are extracted impacted molars. Our team actually is focusing a lot on gingiva, because we found a lot of progenitor and stemness potential in gingiva and oral mucosa. So this is also a very interesting subject, when you can actually acquire stem cells, stemness from the gingiva and mucosa as well. So the imagination is more important than knowledge, for knowledge is limited, but of course we're trying very hard to have the knowledge that we should have in our dental offices, of course. We should ask proper questions, we should use proper materials and know exactly how those materials work, because this is essential for the successful result of our treatment. We should engage patients' own biology, which is very essential. And of course, we want to acknowledge our partners, Solivia, Institute of Advanced Sciences, PPNT in Poznan, and in University in Masaryk, and of course Poznan University is our main area of research and our main partner in this whole oral derived stem cells project. So thank you very much once more for the invitation.
Thank you.
We are very grateful to and happy to share our knowledge with you. So if you have any questions, don't hesitate to ask.
Okay, thank you so much, Dr. Marta, Dr. Bartosz. I'm really glad and thankful for this amazing webinar. It was more than informative for me, and I hope it was the same for the students who were watching us today, and the ones who will watch it in the following days through the social channels of IADS. So let me go through the questions that we have received so far.
Yes.
Okay, so the first question saying that you have mentioned that there will be a chance in the future to use patients' own pulp stem cells for regeneration. Could these also create a potential for making oral stem cells banks where the pulp stem cells are extracted already from maybe deciduous teeth or impacted molars, and then stored until there might come a need for them?
Yeah, that's the idea. That's the purpose as well, to store, especially when we are talking about milk teeth, because we of course know that these stem cells coming from those teeth, they present very high potential. So this is of course a very good idea to store milk teeth, but of course they cannot be completely resorbed. So that's so the situations in which you can store these teeth are, I would say, in part selected, because not all of those teeth will present such a high potential of such a stemness. The idea is that if they are completely resorbed, it might be difficult, but if they are in part resorbed, we are still able to recover those cells from milk teeth. So this is a very good idea to store them for the future application. As you can see from, of course, these were just the examples of some of the studies that are that were already done or are right now happening. But the idea is that some of those therapies could be actually introduced relatively quickly, especially if we're talking about the therapies that don't really include high manipulation of the cells, because if they are highly manipulated, this is sort of a very complicated issue. But if they are not highly manipulated in the laboratory, then it's much easier to introduce them into the practice, and it could be actually possible relatively quickly. So that is, of course, the idea, to store those tissues. Different approaches, you can see different possible approaches. It could be to store teeth, to store tissues, to store cells, cultured cells, different possible options. Although the main idea is, of course, to store them in when they are not really manipulated. So I would say in an original state. For the future application, we already that's already tested, that you can defrost them and they are still working nicely. So the concept is very good and very optimistic.
Yes, in my point of view, the 20th century was the century of the stem cells isolated from the cord blood cells, and the adipose tissue derived cells, stem cells too. But in my point of view, in my opinion, the 21st century will be the century of the dental stem cells and banking of the stem cells. And moreover, we can say also about some exosomes, because exosomes is the future of the stem cells for sure, because we can use the exosomes between several people, without any immunological barrier, and use the exosomes in the practical, preclinical, and clinical studies in the therapies too. That's so that the future of the stem cells are the exosomes, which are isolated, collected from the stem cells too.
Okay, thank you so much, doctors, for this very informative answer. I think we have got one more question here. So the second question is saying that, do you know if any dental schools also teach already stem cell therapy for students?
Do we know dental
schools?
Dental schools.
Yep.
I know that King's College is the pioneer in dental stem cells. So this is, I would say, like a reference point to all of us, I think, involved in stem cells derived from the oral cavity. So to me, I think to us, definitely King's College is the place, the place when it has all started with Professor Paul Sharpie, and they were, I would say, also our inspiration, probably. There are probably other places as well, but I think from the oral cavity, then definitely King's College. We are not that aware of United States. I'm almost sure that there are some places there. But in Europe, it's King's College, probably the reference. I would say the reference research facility, yes, for the research on the oral cavity stem cells. And they were also our inspiration, because also we started a collaboration with the University
of King's College.
Yes, just because they are really the ones that started this whole, I would say, this whole concept and the whole project.
Okay, that's perfect. So the third question we have here is asking about the PRF, which is a platelet-rich fibers that you have mentioned today. So in comparison to PRF, are these surveys showing that stem cells and oral surgery have a better clinical outcome?
It's a different concept, I would say. I mean, I involved it in a presentation because, of course, PRF and concentrated growth factors are working well and are already tested. But this is slightly different approach because in case of PRF, you are using patient's blood, and in our project, we are actually talking about stem cells coming from the oral cavity, and their potential is different because you can imagine that based on the PRF, we can have better healing. It's sort of like blood clot, right? We are sort of stimulating blood clot to better heal. In case of stem cells, the idea and the possibilities are much, I would say, much wider, and we can differentiate them into a completely different cells that can probably also be introduced into completely different areas. For example, they can be differentiated into the neural cells, of course, also to osteoblasts. So there are a lot of different applications, and also the idea is that if you are using patients' own stem cells, you could actually, it was actually in the presentation as well, you could also stimulate, for example, dental pulp to regenerate, or you can, for example, involve them in the treatment of periodontal diseases. So it's a different approach, but of course, in both cases, you are in a way trying to activate patients' own stem cells, and you are trying to do the regeneration, I would say, but it's a different approach. This one is from the blood. In our case, it's from the oral cavity. So if we are talking about, for example, exosomes, so the microvesicles, they actually remember their own origin. So if they come from the oral cavity, they will much better work at this area. If they will come from the bloodstream, it's completely different, right? It's a different approach. Both of those ideas are, of course, part of the, I would say, in a generally biological approach to treatment.
Yes, during my presentation, the first part of the presentation, I show you some genetic basis of the regeneration and the wound healing and vasculature development of the mucosal cells isolated from the pig, porcine back approach mucosal cells. So we have during this presentation we had the ontological expression of the genes in which are involved in the ontological groups responsible for the wound healing exactly. So we have increased possibility to regeneration and the reconstruction of the cells in mucosals too, as compared to the other tissue of our organism, I think.
So if you ask if the question would be what is the difference, it's a very big difference. But on the other hand, the PRF is already very well tested in clinic, right, in practice. So we are talking about something that's already been in a way introduced into the practice. In case of our research, it's more like we we really think it will be introduced into the practice, but it still requires more testing, right? And we need to move on to the clinical part. And PRF, I would say, is used very often as a scaffold for something, right? As a scaffold for healing or for regeneration. So this is like a base for regeneration, which is of course a very good option. It doesn't work all the time. It can be observed that it doesn't work in each case. Difficult to say sometimes what is the reason why it's not that successful in all of the therapies. For example, in peri-implantitis, some of the studies will show you that it's effective. In some patients, it's not really effective. And in those patients, you need something more. So the idea is also that in case of more research on stem cells, the idea would be also to have some bone grafts that you could actually really culture patients' own cells and differentiate them into some particular cells and then use them for grafts or for personalized dentistry or medicine. So it could go much further. But of course, PRF is something that we use on daily basis. So it is something tested in a way. And in case of the other concept that we are discussing now, it still requires testing, and it requires some time to be introduced into the practice, especially when we are talking about legislation. That's why we are very often coming back to exosomes, because it looks like they do not require such a high manipulation, so they could be relatively quickly introduced as a stimulator for the regeneration processes.
As you can see in our presentation, we have a lot of in vitro experiments, in vitro results from our research, from our lab, based on the IMA model, based on the Porcine model, and the human model too, including the gingiva, including the mucosa, including the dental pulp, porcine buccal pouch mucosal cells, and many others. So we have a lot of in vitro results, but now we are starting with the banking of the cells. That's why we are collaborating with Celivia, which has their own bank, and then we can start banking of the cells for the future direction and future application in the preclinical, clinical study, and to help, of course, the patients in the future. Yeah.
Yes, thank you so much, Dr. Bartosz and Dr. Marta, for this amazing presentation today. I have to acknowledge that it was really informative. For my colleagues who were watching, they sent me some feedback about it, so we are really happy for that, and we would like to thank you again for accepting our invitation and for your time today. And I would like to thank all of our viewers who have been watching us through the social channels of IADS, either the YouTube or the Facebook. Thanks all, and have a nice day.
Thank you very much.
Thank you very much. Have a good day.